Okay, thank you very much for the introduction. Can everyone hear me at the back? Yep, excellent. So my name's Laura McGregor. I'm here from Schoenberg Analytical, as you can see on the slide set. So first of all, I just want to introduce the company and who we are in relation to Marks International. So both companies are actually part of the Schoenberg Analytics group. So while Marks is focusing on the sample preparation side of technology, as I'm sure you're all aware, I think there are many people in the room using thermal desorption. Schoenberg are more focused on the separation and the detection, but we are sister companies, so we're working very closely together. So as I just said, I know a lot of you are using thermal desorption already, so I'm not going to bore you in this presentation by going through what it is and how it works. Instead, I'd like to focus on some of the ways that you can use TD to be able to get sample security and quality control. Then I'd like to look at samples beyond breath as well. So how you can look for biomarkers and other types of sample. So I think we've seen over the excellent presentations over the last day and a half that breath samples are incredibly complex. There are over 800 different volatiles that have already been reported in literature. So many different sample classes, wide-ranging concentrations. Are we sure that they're all biomarkers? Which ones are really biomarkers? And there's a lack of consensus about this. So altogether, this makes it quite a challenging area of research. Regardless, in the past 10 years, there's been a huge influx in the number of clinical trials that actually involve breath analysis, and specifically breath biomarker analysis. So why has it not progressed to the next stage? Well, I think from all of the presentations we've heard so far, there is a need for standardization and for quality control to be able to move to the next level. So just to briefly touch on the challenges involved, we've obviously got a range of different sampling options to collect the breath. They have low sample volumes and very high humidity, which can cause issues when you're looking at GCMS separation. The samples, as I've shown already, are very complex, and if the compounds are in trace levels, you need very sensitive detection. And a really important part is the reproducibility, not only for the sampling device, but also for the full analytical system. So as we've seen already, the first standardized breath sampling device is the receiver to be able to sample specific volumes and fractions of breath. But you've also got to remember the rest of the analytical system as well, and ensure that you're not looking at compounds that are from consumables and introduced within the system itself rather from the breath sample. So you have to ensure that there's minimal chemical background on all parts of the equipment, from consumables all the way through the TD, GC, and MS. So this just summarizes some of the variability issues that can happen within the system. We know already that there are many external factors that can contribute, but we're going to focus here on this side with the storage, contamination, quality control aspects. So I just want to take a look at a few problems and solutions when it comes to the use of thermal desorption and why it's been stated in many presentations so far that thermal desorption and GCMS is the gold standard for breath analysis. So of course, you need to be able to collect breath from multiple different sites, transport those samples to the lab. You may need to store them as well. With thermal desorption tubes, they're very compact. They can easily be capped to seal them for transport and for storage to ensure you've got sample security. Breath is saturated with water vapor. The sorbent tubes can be selected to retain as little water as possible, and we can perform dry purging of the tubes and the focusing trap prior to analysis to remove further water. And you saw in the Owlstone video earlier the use of the TC20 conditioning system. With that system, you can actually perform offline dry purging as well for up to 20 tubes at a time. So rather than having the dry purging within a sequence, you can do it offline in the TC20. You've got to be able to do untargeted analysis because we don't know what the biomarkers of interest are going to be. And they can be at trace levels. So within thermal desorption, using a cryogen-free trap means that you're going to be able to focus the analytes and pre-concentrate the sample prior to GCMS. So you can increase the sensitivity. I think we all know that clinical samples are invaluable. So being able to recollect a portion of your sample onto a clean sorbent tube for repeat analysis is invaluable in this case. You can recollect for analysis under different experimental conditions if you were not sure of what you should have used in your GC and MS conditions, or for repeat analysis to get replicate data. Because the more data that we have, the more confident we can be in the results. And when you have lengthy clinical studies, you're transporting tubes and samples between lab and the sampling site a lot, then being able to add internal standards to the tubes and to the focusing trap to ensure quality control is very important. So just to go back to the sample security aspect, the sorbent tubes can be sealed at point of use with the brass caps to ensure that you're not going to get any contamination going into the tube or any of your sample leaving the tube. And then when they're waiting in an autosampler for analysis, they can have the diff lock caps fitted so that, again, if you have a long sequence waiting for analysis, you're still not going to get contamination going into the tubes. In terms of the audit trail, we have the barcoding on tubes, and we can provide RFID tags to be able to track where tubes have been, how they've been sampled, and what's been on the tubes and how many times they've been used. And of course, thermal desorption is compatible with a range of different samplers, and throughout the talks in this conference, you've seen various different options. So George already mentioned about standardization and the metabolomics type approach to standards. And the one thing that is missing on this list is, of course, the pooled samples, because it is so difficult to do this for breath analysis. The closest that you can do is to create standards in a humid gas and try to create a breath surrogate of sorts. But we can ensure that the analytes are definitely of biological origin by running system blanks. We can run suitability samples, so load the TD tube with standards and use this as a quality control check to ensure that the system is working to the set level before you start using your precious biological samples. And of course, we can add in internal standards, again, to ensure quality control. And the one thing at the end here that we've mentioned is that maybe the next step would be to have a standard reference material generated that is created by a third party so that everyone in the field of breath analysis would be able to use that standard reference material and validate their systems accordingly. So something generated by, say, the National Physical Laboratory. So there are numerous ways to get standards onto the tubes to do the quality control. And I'm just going to look at a couple of ways of doing automated standard addition to either blank tubes, to sampled tubes, or directly onto the focusing trap. So in the Marks TD systems, you have within the thermal desorber a way to introduce standard or internal standards into the loop, and then flush the loop either onto a sample tube or directly onto the focusing trap. So there are two options there for introducing standards. Oh, sorry. What we have done at SEPSOLV as well is to take this a step further, so that if you have a high loading standard on one tube, you can use the recollect 10 to be able to split that sample onto 10 tubes at once. So then you would have 10 tubes that have the same loading of standard, which could then be run in sequences at set time periods as a quality control check. So it means that you would have the same levels of components. Here's an example of some standards that were introduced, and quickly have created 10 tubes with the same quality control standard. We know that it's also quite difficult sometimes to get all of your standards in gas form. So one of the other things that we have developed is a way to introduce liquid standards onto the sorbent tubes. So here, we've created bespoke trays for the sample preparation robot so that they will hold the sorbent tubes, and then we can use the sample prep robot to spike the tubes automatically with liquid standard. So you can see the workflow here, where the tubes are held in the bespoke tray. Then the liquid syringe tool will load the standards onto the tubes before switching to the headspace syringe to purge the tube with nitrogen to eliminate any of the solvent. So being able to accommodate over 200 tubes on one autosampler means that you could very quickly spike multiple tubes with standards. So that's some of the sampling side. Now, when it comes to identification of biomarkers, how can you be sure that your compound is actually what you say it is? We've heard already that one option for this is to do high-resolution MS, but the part I want to focus on here is increasing separation to be more confident about your identification. So the challenge within the breath samples is that you might have coalitions happening, which can hinder your identification. Deconvolution, after all, is just an algorithm. Can you be confident that you're getting the right answer? With GC-GC, you can get improved separation of your sample. So instead of having the coalitions, you separate out peaks. So those who aren't familiar with GC-GC, here each colored spot is representing a peak with the height of the peak represented by the color intensity. So the more red, the larger the peak, the more intense it is. So instead of having one column with one stationary phase to separate your sample, in GC-GC you have two. So instead of getting one peak separated out here, you can see that in the GC-GC plot, we have two peaks that would have perfectly correlated in a one-dimensional separation. But by separating them out, you can get cleaner spectra to be more confident about identification. So just to show this in practice, we have here a brief example of the peppermint experiment, where we measured a breath sample before a peppermint capsule was consumed and 30 minutes after. And I just want to draw your attention to the boxed area here, because this is where the key terpenes are eluting, the ones that are changing after the consumption of the peppermint capsule. And you can see that in this second dimension, there are numerous polar compounds here that have been separated out that would have co-eluted with the compounds in the boxed region. But with GC-GC, you've separated all of that out, and then you can zoom into that area of interest with the terpenes and be confident about your identifications. Another technique that we've introduced to add confidence is to acquire tandem ionization. So traditionally, in GCMS, you would acquire 70 eV electron ionization. But what we do with our benchtop instrument is to acquire both 70 eV and softer ionization energies in the same run. So you switch rapidly between the two and collect both data sets in one analysis. So it means you have a second set of spectra to confirm the identity of the compound. And this is really important when it comes to things like isomers, because they can have very similar 70 eV spectra, but when you look at the soft EI, you can see differences that help you to be confident you're identifying the right compound. So just to show a few examples, it means that in any GC or GC-GC run, you would be able to develop two sets of MS data. And you see here for 2-pentylfuran, the increase that we're getting in the molecular ion there. So you're going to be able to pick that out of a complex sample more easily using the 138 ion. And a couple of extra examples here. We see octanol, where you're increasing some of the higher end ions in the spectrum and minimizing the lower end. And again, for ethylphenol, here the molecular ion becomes the base peak. So it allows you to develop a library with 70 eV data as well as soft EI data for that extra confidence. And when you combine that with things like retention indices, as well as the retention times in first and second dimensions, you've got additional confirmation that you're identifying the correct compounds. So the last part that I want to look at is beyond breath. So what about all of the other types of sampling that we could do to look for biomarkers? Now, here I want to introduce the new Sentry system, where not only can you do tube-based thermal desorption in the same manner that you currently do, you can also automate a range of other sampling systems to be able to look at different types of sample, like urine or skin, sweat, all of these different biological samples. So within the Sentry, it's using the Mark's trapping technology to expand the performance of traditional sampling techniques. So we can do classical headspace analysis, but also headspace and trap to be able to provide focusing of the analytes and enrichment of the sample. The same is true for SPEMI. You can do your classical SPEMI or SPEMI trap, where, again, the sample is sent to the trap before GCMS for focusing, so you get improved sensitivity and you can enrich the sample on the trap. So multiple SPEMI samples can be sent to the same trap before it is fired to the GCMS. As I said, you can also do the tube-based thermal desorption using a 50-tube autosampler. And finally, high-sorb sorbtive extraction. So this is working in a similar principle to SPEME, but on a much larger scale. So a much larger amount of phase on the sorbtive extraction probe. So it just means that because you're using the trap, you can benefit from all of these different features here. Being able to do the sample recollection from any of the different modes. So SPEME can then be done with recollection of the sample. You get improved chromatography, sharper peak shape. You can get better selectivity because you've now got a trap that you can purge. You can enrich the sample onto one trap, so multiple vials can be sampled onto one trap. And you've got the flexibility to use lots of different modes, all in an automated system. So I just want to show a couple of examples of some of the work that's been done in this type of sampling system. And here we have a GC-GC TOF color plot for skin volatiles. So this was done using thin film SPEME on the skin, and you can see the wide range of components that have been separated out here. There's actually 800 different peaks separated. And we've got a region here for fatty acids, but just to show you, we've got alkanes eluting at the bottom of the second dimension here, but all the way to the top, we have methylfuranone. So we're utilizing that entire space to separate out all the components. And I've just pointed out the DET here because it's one of the most common insecticides, so present in a lot of insect repellent. And I think our candidate here had been using that before sampling. Then finally, we have the high-sorb sorbtive extraction. So you can see the probes are very robust, not like SPEME fibers that are prone to snapping very easily. And you can perform headspace of the sample as well as immersive extraction. So I just want to take a look at the workflow here because the sorbtive extraction until now hasn't been fully automated. But within the Sentry, the robotic arm will take the probe to the vial to allow for agitation and heating while the sampling is done. It then takes the probe to a wash station where it will rinse it free of any matrix and dry the probe. Then it will desorb the probe in the inlet. The analytes are sent to the trap for analysis by GCMS. And since they are wider than your average SPEME fiber needle, we do have this capping tool here, which can plug the vials to ensure that none of your sample is lost to the lab air or nothing from the lab gets into the sample. So just to show an example of this in action, this was conducted by the team at ICL, where they did a preliminary study using the high-sorb probes. And you can just see in the expanded regions here all of the different compound and chemical class types here that were able to be sampled using a high-sorb. And in the preliminary study, they looked at 100 different participants and were able to get 90% correct discrimination between the esophagogastric cancer patients and the control set. So as a preliminary study goes, that's looking pretty promising for the high-sorb probes for urine analysis. So in this talk, I've showed the thermal desorption that can be utilized with GCMS platforms, but we can now incorporate things like the Sentry to expand out from breath analysis, and then a step beyond to our discovery platform where we can see as much information as possible in the sample using GC-GC TOF in combination with the Sentry. So just to summarize, I think we've seen already that TD GCMS is the gold standard technique for breath, and hopefully with some of the quality control aspects that I've showed here, you can see why this is the case. And with the soft ionization, using tandem ionization, as well as using the separation of GC-GC, you can enhance the confidence and make sure you're identifying what you say you're identifying. So thank you very much for listening. The change in the schedule is quite beneficial to me because now I can hopefully direct any of your difficult questions to my colleagues at our booth, but I'd be happy to take any easy ones now. Thank you.